Hello everyone, myself Nisha. In this video I am going to explain about case study on Frankia species. Introduction. Frankia species is a genus of nitrogen fixing actinomycete bacteria known for their remarkable symbiotic relationship with specific plants. These interactions are ecologically and agriculturally significant as they enable plants to thrive in nutrient poor soils. The genus Frankia was originally named by Georgen Bunchorst in 1886 to honor the German biologist Albert Bernhard Frank. Characteristics of Frankia species. Gram's classification. Gram-positive actinobacteria. Taxonomy. Frankia is a genus within the actinomycete group of bacteria belonging to this family Frankiaceae. Morphology. Frankia species exhibits filamentous growth and can form specialized structures known as vesicles and sporangiae. Nitrogen fixation. One of the key features of Frankia is its ability to fix atmospheric nitrogen, converting it into a form that plants can utilize for growth. Optimum temperature. Optimum temperature is 30 degrees Celsius. Sporangia. Sporangia are another important structure formed by Frankia. These structures contain spores that can be important for the reproduction and dissemination of Frankia. Optimum pH. Capable of growing at pH level between 6.5 to 7. Genetic diversity. Frankia is a genetically diverse genus with various strains that may exhibit differences in their genetic makeup and symbiotic capabilities. Motility. Non-motile. Actinorhizal symbiosis. Frankia species forms mutualistic symbiotic relationship with specific plants known as actinorhizal plants. This is a key characteristic as it enables these plants to thrive in poor nutrients. Tolerance to extreme conditions. Some Frankia strains are known to tolerate extreme conditions including high salinity and low pH, making them adaptable to various environmental natures. Respiration. Microaerophilic in nature. Generation time. Very slow growth rate 24 hours to 48 hours. Capsule and endospores absent. Ecological importance. Nitrogen fixation. One of the most important significant ecological contributions of Frankia species is its capacity to fix atmospheric nitrogen. It converts atmospheric nitrogen gas into ammonia, which is a form of nitrogen that plants can readily absorb and use for growth. This is crucial in ecosystems with nitrogen poor soils, as it provides a source of essential nutrients for plant growth. Reforestation. The ability of Frankia species to enhance the growth of actinorhizal plants makes it valuable in reforestation efforts in degraded or marginal lands. Enabling plant adaptation. Frankia species allows certain actinorhizal plants to thrive in nutrient-poor soils, where nitrogen availability is a limiting factor. These plants have evolved to form mutualistic association with Frankia species, and these adaptations enables them to colonize a broader range of ecological nations. Soil improvement. By enhancing soil fertility through nitrogen fixation, Frankia species contributes to the overall health and sustainability of ecosystems. It enriches the soil with nitrogen, making it more suitable for plant growth and in turn supporting the entire ecosystem. Biodiversity. The presence of actinorhizal plants facilitated by Frankia species contributes to biodiversity in various ecosystems. These plants can serve as habitats and food source for range of animals including insects and mammals. Carbon sequestration. Actinorhizal plants with the help of Frankia species can capture carbon dioxide from the atmosphere and store it in their biomass and in the soil. This aids in carbon sequestration, an essential component of mitigating climate change. Habitat restoration. In ecosystems affected by disturbances such as wildfires, erosion, or land degradation, actinorhizal plants supported by Frankia species can play a vital role in soil stabilization and habitat restoration. They are often used in ecological restoration projects. Isolations of Frankia species. First, sample collection. Collect root nodules or soil samples from the root zone of known actinorhizal plants such as Ulmus or Casuarina, where Frankia species is likely to be present. Surface sterilization. To eliminate surface contaminants, carefully sterile the samples by washing them in a series of steriling agents and rinsing with sterile water. Crushing and homogenization. Crushed root nodules or mixed soil samples to release Frankia species cells into a sterile buffer or solution. Dilution and plating. Dilute the homogenized sample and plate it on a suitable culture medium. Frankia species is often isolated using various complex media such as modified Strickland medium or modified crop medium. Incubation. Incubate the plates under appropriate conditions, example at room temperature or slightly above, for several weeks, as Frankia species can have a slow growth rate. Colony selection. Once colonies appears on the plates, select individual colonies and subculture them on fresh medium to obtain pure isolates. Identification of Frankia species. Morphological characteristics. Microscopy can reveal the filamentous branching hyphae of Frankia species, including the formation of vesicles and sporangia. These structures are key morphological characteristics. Molecular techniques. DNA-based methods are commonly used for accurate identification. This involves polymerase chain reaction (PCR). Specific primers are used to amplified regions of the Frankia species genome, such as the 16S or RNA gene. Sequencing. The amplified DNA can be sequenced and compared to known Frankia sequences. Phylogenetic analysis. The obtained sequences can be used to construct phylogenetic trees to determine the relationship of the isolated Frankia species to known strains and species. Physiological and biochemical characteristics. Testing for physiological and biochemical traits such as nitrogen fixation ability, growth in various culture conditions, and enzyme production can provide further identification information. Host plant compatibility. If the identifier of the host plant is known, its compatibility with the isolated Frankia can be a useful clue for identification. Genome analysis. Advanced techniques used like whole genome sequencing can provide comprehensive information about the isolated Frankia species, aiding in its identification. Recent research. Medicinal applications. Researchers are exploring the potential health benefits of components produced by Frankia species, such as antibiotics and bioactive molecules. Biotechnology. Frankia species is being investigated for its biotechnological application, including the productions of bioactive components, enzymes, and biodegradable materials. Genetic studies. Advances in genetic research are shedding light on the diversity of Frankia strains and their genes involved in nitrogen fixation. Environmental applications. Frankia species is being studied for its potential in environmental remediation, such as the bioremediations of polluted soils. Thank you.